Okay, so I'm thinking we're ready to go. Yes?
Yes.
Okay. Welcome to our second faculty research symposium for this semester. As we've talked about before, we're doing faculty research symposium a little bit differently this year in that we have panels talking about how their work centers around a topic rather than doing one individual presenter for the whole time. So today's panel is single case design. And if you were able to join us for Dr. Zhang's Lunch and Learn earlier in the month, you know that single case design is a really interesting intervention research design that can be used to ask a variety of clinical questions. It is not to be confused with a case study. This is a rigorous design that requires replication and helps prove effects on clinical populations, but also can be done with a smaller sample size. But I think you'll probably hear from the panelists that that doesn't necessarily make it easier because there's a lot more work involved on the other side. So today we've got Dr. Danielle Brimo presenting, Dr. Adam King, and Dr. Ahmed Rivera Campos. So I don't know how many of them you can see from online, but you guys can certainly see them if you're in person. Yeah, a little bit. Okay. So what we're going to do is have each of them present on a study that they have recently completed or is ongoing using this methodology. And then we're going to open this up to a true panel style presentation where we can take questions. We can try to take questions online. We can definitely take questions in person. And I will attempt to act as the discussant for this work. So we can kind of think about how single case design can be used across all of our different health professions in Harris College. So with that, I will hand over to Dr. Brimo.
And you'll click for me?
Sure. If I can figure out how. This is like my everyday struggle. There we go.
So thank you for those who are in person and online. I can see you all if I turn around. So like Emily said, I'm Danielle Brimo. I'm an associate professor in the Communication Sciences and Disorders Department. I'm also the director of the SAIL Lab. It's the Syntax Assessment Intervention Literacy Lab. I've been plugging that more often now to get it out there. And I wanted to share first really the work that I do as part of the SAIL Lab is I'm really interested in examining children's spoken language as it relates to their written language. It's a very broad kind of overview, but specifically, I'm really interested in looking at children's syntax production, particularly their complex syntax production. Oh yeah, I don't really talk loud. And looking at how that affects children's reading development, their writing development, and their reading comprehension. More recently, I've been looking at children's spelling errors, especially among kids. So I really like the school age population. So second, third, fourth, fifth graders are really my jam.
This work.
Perfect.
Oh, I didn't have it on.
No,
No, it's on.
is that better?
Yes.
Wonderful. Do you want me to start over?
No.
No. Really like school age kids, and I really like kids with learning disabilities, so developmental language disorder or specific language impairment, and kids with dyslexia. So more recently, I've started to look at spelling and the language-based skills of that population and how we can look at their language errors within their spelling errors. The project I'm going to present today, the single case design study, it was part of a graduate thesis. So Miguel Garcia Salas, he graduated last spring. He is currently now a PhD student at Florida State University. So that's exciting. How do I click? Oh, click. So part of this study, so the purpose of this study, we were interested in understanding whether parents could learn or we could teach them reading strategies. This came from a discussion earlier on and a couple of graduate thesis projects before that. related to parents being like, children are assigned homework and they're assigned to read 20 minutes at home. And so, you know, I think about kids with dyslexia or kids with language impairment and how they may struggle with that. And one kid in particular that I worked with, he would just look at a Pokemon book and look at the pictures for 20 minutes. So I thought, can we intersect some of the work that within the field, training parents to use specific strategies with their kids, training them to like sit next to them, and do some specific training to see if that benefits. So we recruited 2 parent-child dyads. They were both in the 2nd grade going into 3rd grade. Both kids had dyslexia. Mainly, yeah, so both kids had dyslexia. We gave a battery of tests to the adult participant. So we were more interested in the adult participant than the child, but the child was part of the intervention. We gave an adult like language battery. I also tested the adults phonological awareness, which is their manipulation of sound, their reading as well. And then what we found, so one parent, and it's parent-child dyad 2, that parent self-disclosed that she had dyslexia, and so did her husband, and so did her child. And then parent-child dyad 1 did not have any family history. However, when we completed that battery of tests, both parents fell in this vulnerability of language or learning disability. And so parent 1 wasn't quite aware, but parent 2 was well aware that she had dyslexia. So we picked 3 reading strategies to teach the parents. This was based off of literature in this field, looking at what effective strategies were already available. And these were ones that like independent research taught the kids themselves. It wasn't a parent training model. So we were really looking, could we take these reading strategies that we know work to improve a child's reading and teach them to a parent. So the sounding out and blending one was teaching them to take a look at a word, sound out each letter in the word, say their name, and then blend it together. The meaning-based one was a little bit more language-based in that we would, if the child made a reading error, we would tell the parent to say, ask them a general question like, does that make sense? What do you think that word means? So a little bit more open-ended questions related to the child's reading error. And then the dividing the word strategy was a strategy related to picking out larger units within the word. So a word like running, we would have the parent put their hand over ing and show them run, and then put their hand over run and show them ing, and then blend those larger chunk units together. So those were the 3 strategies. The design for this study, so the single case design, which is probably the most important part of this presentation, was a multiple baseline across behaviors. We initially wanted to do it across participants. COVID recruitment is always a struggle. This was a graduate thesis, so I couldn't recruit forever. He needed to graduate. So we ended up changing it a little bit because we only had 2 participants agree to participate in the study. So we had these 3 behaviors, and then what we did in terms of the design, and it'll be a little clearer once I show you the 3 graphs, is that we changed the order in which we taught each strategy to the parent. So we could also look across both participants as well, because each intervention reading strategy was taught at a different time. So like off the top of my head, I think that like parent dyad 1, we did that order. And parent dyad 2, I think we started with dividing a word, then we went to sounding out, and then we did meaning based. So the way that we designed it was we had 5 sessions for baseline, which is, I think, a good standard for getting a stable baseline. 3 is like the minimum, but 5 allows you just to make sure that there's no increase in the baseline. So we plan for 5 baseline sessions, and then for each target, each reading strategy, we plan for 5 teaching intervention strategies. Literature said that kids responded to it fairly quickly, so we thought, you know, if we could get some change within those 5 sessions, that would be good. That also kept his intervention limited. So again, if maybe this wasn't a student project, I could go 6 sessions or 8 sessions for the reading strategy, but this was a 20 session long study. And so, 6 weeks, 5-6 weeks with the parents. So we also wanted to make it socially, viable for the parents as well, because they were, spending a good deal of time with us. Just to let you know that, so baseline, when we collected baseline, we did not provide the intervention at all. We just told the parent, hey, read for 15 minutes. We just recorded it. So this did happen on Zoom. It wasn't initially supposed to happen on Zoom, but due to COVID, this was basically like the, planned his project all the way, and then we had to change it last minute. So all of this, we switched it to looking at whether we could train parents through telecommunication or through Zoom. And so the first 15 minutes, so the baseline session was just read with your child. Thanks, we'll see you next session. We tried to do those consecutively. We didn't have to, it was just based off of the parents' schedules pretty much. So if they said we could do it 3 times a week this week, great. But when we got to the intervention, it was always 2 days a week. And then how we organized the intervention is we always did data collection the first 15 minutes. So the first part of the session was the parent reading with their child. We didn't give them any directions. We just said, read as you normally would, in hopes that once we started the intervention, we would see more of those parent behaviors that we wanted to see in that first part of the intervention. And then the rest, it was about an hour. The rest of the time was devoted to the actual intervention. So we use the teach model coach review model of parent training, which in our field, and I don't know, you would know more a little bit, if it's early intervention in general, but in our field, it's mainly used for younger kids and to teach them language-based strategies. So we're really taking this model of parent training and applying it to more reading-based skills to see if it would change. Go ahead. Am I? I'm okay. 4 minutes.
Yeah, 4 minutes maybe.
Okay. All right. So this is parent-child diet 1. And because we did it across behavior, that's the way that I laid out the graph. So everything on the left side of the vertical line is our baseline, and it's staggered. So like each intervention I weighted. So one intervention was implemented at one time. So you can see for like parent 1, her baseline kind of went up, but it stayed pretty much so stable. Same thing with the second strategy, and you guys can't see it, but their names are covering which strategy. I mean, the pictures are covering which strategy each graph was at. So session is at the bottom, and then percentage of use. Oh, there you go. Percentage of use is on the right side, on the left side, and then that was the 3 strategies. So for this parent, we implemented the sounding out and blending strategy for 5 sessions, and what we were looking for is growth right after that line. The same thing for the meaning-based strategy, and then the dividing a word. And this is where I have to go to my notes. So what I looked at, and you can go to the second one so they can just see it as well. So same thing for this parent, although the order of the intervention strategy shifted. I look at level, and I don't know in this, I didn't get to go to the single case training, so hopefully this is correct. So level and overlap looks at, so visually, is the baseline different than the intervention phase? So if you kind of put like a horizontal line on that, like a top point, that 10% point on that first line, you could definitely see that there was a level shift, that there was an increase after the intervention was implemented. So I look at level, a popular one is more percent of non-overlapping data. So that's looking at the number of data points that are different in the during the intervention phase than the baseline phase. So for this parent, what we concluded is that for the dividing a word and the meaning base, we saw an effect. We saw a good immediate effect for the dividing the word strategy. She kind of trailed off after that because I think other strategies were implemented. And then you kind of see the same thing. We did see some use of the meaning-based one, and then there was a steep decline. This is the parent that had dyslexia. And so for the sounding out and blending, And for anybody who knows about dyslexia, that's a particularly hard skill. So for that parent, she didn't like it. It was hard for her. And you can see that we didn't find an effect. So in terms of single case, we wouldn't really say that this, because I only had 3 behaviors, I saw an effect for 2 of the 3 behaviors and not the third middle one. If you go back, I can quickly, hopefully I have like a minute. For this parent, we saw an effect for the sounding out strategy, the meaning-based one, little bit, but not the dividing a word strategy. So again, for this parent across behavior, we really didn't see an effect. Now, what I did in the paper, and hopefully these reviewers buy it, I'm hoping.
It's a legitimate thing you can do.
I think so, is I then, because I presented each intervention in a different order, I was able to compare across the 2 participants, and they happened to show an effect across all three strategies. So what I tried to do in the paper is to argue, I didn't see an effect across behavior for each individual, but when I looked at both, we did see an effect across. And so I could say that there was a functional relation between my intervention and these parents' behaviors. The end, I think. Okay. Yeah. And I can talk a little bit more about that if you guys have questions about how I tried to argue. And what's nice, really, to flood single case is that you get to look at each individual family or each parent. So like, I really talked a lot about in my discussion about this particular, like we like parent training, but for this mom that had dyslexia, this strategy wasn't, she needed something more than just me showing her how to do it. So that's my plug for single case.
Go ahead and get everything set up with the microphone. So now we're going to switch to our next presenter, who will give us a different view of using this design.
Right. Stand a little bit offside on there. That projector's in my height range. Thanks for coming today and joining us for this. My name is Ahmed. I'm from Department of Kinesiology. This work comes out of stuff that we do in our motor behavior lab, where we're taking a look at how individuals control movement, learn movements, and we take that through several different aspects of performance enhancement, injury prevention, and rehabilitation. We've got a variety of tools that we're using to look at how individuals perform our motor skills. The one I'm going to focus on today is on that left-hand side. That's called a force plate that we can measure center of pressure and postural sway. But we also have the ability to do some motion capture and measure some muscle activity that's going on from the neurological level to understand neurophysiology components of movement skills. Next slide. The paper I'm going to talk on, actually thought I might get kicked out of here today with Emily's introduction, because it is a case study and not a true single case design, but it does lead into, I think, two lessons to be learned for all of us as we're kind of considering options and potential for how we conduct our research. And I did use that title intentionally because stumbled upon this. Not in my background, not my training, but I think as researchers, we know it's important to have a key eye on our data collection procedures and things that are going on in that environment. We teach our students to do that. We lose opportunities at times of doing that, myself very guilty of that. But this paper that came out of here for recovery from a concussion was from that skill in seeing what's going on with our participants. One, having a little bit knowledge of that individual being within our department and being recruited from there. And then just being aware of an opportunity to look at a question that we were interested in. The main topic that we were kind of guiding towards is postural control. Yeah, the next one. So our original plan in paper that came out of this was focused on young, healthy individuals who would stand in an upright position under these different conditions where we would manipulate the slope of the surface that they're standing on. In that left-hand image down there, we see somebody in this plantar flexion position. We can change that angle. We can also move it into a plantar or inclined position. And they have to try and stand as still as they can. We put it on top of that force plate in the upper right image that I mentioned before. And this measures how much their body sways. Even though we're telling them to stand still, they do not stand still. The trajectories that we see on the two graphs there are the motion of our body in the medial lateral or side-to-side direction. That's kind of plotted on the x-axis. And then on the y direction is our forward and backwards motion that happens as we're standing upright. And it is a small amount. We're looking at that units being millimeters, but it's also informative of how our body organizes our ability to stand upright. The image on the left-hand side is in that eyes closed condition, whereas the image on the right-hand side is in eyes closed condition. So we change, not having that visual feedback or correction causes us to increase our sway. So the slope is 1 degree or 1 manipulation of postural difficulty. The availability of vision is another one that happens in terms of postural difficulty. And then we're also looking at some like short and long trials, but that was kind of far beyond that. And what happened, we basically had we were collecting data, and an individual had been a participant for us. And then a week later, we found out that he had sustained a concussion playing racquetball in the rec center. Fairly severe concussion. It was diagnosed by a doctor, had some pretty intense symptoms in terms of visual disturbance, movement disturbances, sensitivity to light, sleep disturbances happening. So kind of the classic concussion symptoms that you would see with that. And that's where we kind of saw the opportunity of taking this to a injury situation of what's the recovery trajectory. And there's been some work out on that front, but this was an opportunity for us to look at it in a little bit different way, using some postural difficulty conditions to be able to evaluate that. Yeah, you can go to the next slide with some of the data there. So on those squiggly trajectories that I was showing you, there's a number of dependent variables that we can measure from that. One is the amount of sway that an individual has. That is the standard deviation measurement that we have on the y-axis, and we broke that down into medial, lateral, and forward and backwards. It tells us some different characteristics of how people are able to maintain balance. Given that we had another set of healthy individuals that hadn't fortunately sustained a concussion, we used those as some controls, and we extended that as a horizontal line across there as our reference point. So we weren't really using the statistical design of what a single case approach is, but I'll get more to that a little bit later on why that's been helpful for me. And then the triangles that we see on there are our concussed individual, and we brought him back at 1, 2, 3, and 4 weeks after he had sustained his concussion, and ran him through the test that we were running. And one thing that comes out from this measurement of our standard deviation is it seems to be that this individual is returning towards baseline and kind of at the level of our controls when we look at this 28 days post concussion. His symptoms completely resolved after about 3 weeks. Still had some minor symptoms that he was experiencing at like 2 or 3 weeks, but from a behavioral standpoint, symptoms were completely resolved. So this measures one of the common ones that are used for evaluating postural control. On the next slide is another measurement that we are using that is a nonlinear approach that looks at the structure and patterns that happen within those center pressure trajectories, or as I refer to them, those squiggly lines that we saw earlier. There are patterns that are in there that we can detect and quantify. And what we see with this individual, and the point that we made in the paper, was after 28 days from his concussion, there were some lingering effects of postural impairments that were going on with this individual that were masked on that other measurement that we were looking at. So if you want to click there, there's kind of some key finding things that come out of there. One, we knew that there was, yeah, if you could move, help me remind me of what my findings were, because I can't read that far in the back.
Okay.
So that we knew, and this is further evidence that the manipulations that we were doing altered postural control. That was kind of given, and we knew that. But the points in terms of the concussion that we were making with our paper was that there were some persistent effects, which have large implications for those individuals that might be returning to sport or activity in terms of sustaining another concussion, which can lead to even greater consequences than the initial event. The other part that we made in that paper was that it's kind of dependent on the variable that we're looking at, and some measures might seem to be that the individual's recovered, whereas some of our other measurements that give us different pieces of information about how we control our posture tell a little bit different story. A little bit back to that initial point, more rigor and further follow-up needs to happen on this front to kind of validate that interpretation, which again, we did argue about that in our discussion paper, but that was kind of the paper that connected me to the single study design. And again, back to that point about being aware of our situations and looking for opportunities to answer questions that are relevant for us. So I think that was kind of one lesson that came out of there and was able to get that publication. The second lesson is really moving to a new project. We talked, Danielle and Ahmed are going to talk about, population groups. I've worked with young, healthy individuals since I started grad school and have benefited from using young, healthy individuals because we have access to them being on college campus. But we had a project in mind that would move into a Parkinson's disease population, and working with those individuals, obviously access is a little bit limited for there, and being aware of this case design, knowing that other people at the college have used a single study design, we are able to use some of that information to build a study around what we're calling neuromotor function within the Parkinson's population. So we are using this, actually one more slide. We are using this single subject design in the vein of what should be done and how our statistics should be run with a small cohort group that are really right now going through some multiple baseline testing. We are using the minimum 3 baseline testings that are happening, but we also have this level. We are trying to be careful, yes. And you're going to be even more challenging with a variable group such as Parkinson's that are going to happen. But we are collecting a handful, 7 to 8 subjects rather than a single one to help with a little bit of the robustness. But they all are going through this single case design where we are doing that multiple baseline testing. We're incorporating an intervention. No, we are not knocking them out. They're doing an exercise class that revolves around punching out Parkinson's, and then we're tracking them longitudinally over the next year. And so similar to the plots that Danielle was showing in terms of flatline intervention being incorporated and changes post, we're using that design with our study that's being collected right now to evaluate a variety of components around neuromotor function with Dr. Parr in the kinesiology lab, Dr. Porter, and then also with Dean Watts working on several different things of that. But that's kind of what got me into there, which again opens another avenue and opportunity that 2 or 3 years ago probably wouldn't have been aware of or been in my toolbox as a researcher. So I benefited and learned a new approach, which I think all of us can appreciate as lifelong learners and finding opportunities to address questions through ways that we might not typically approach it on. But I'll stop there and we'll turn it over.
Awesome. Okay. And our last presenter today will be Dr. Ahmed Rivera Campos. I'm going to have him come over here once he gets the microphone and tell us about his study. You want this? Here, here. I don't mind.
Hello, everybody. So today, since data collection and group design is so different than single case design, I decided to approach this as describing how I gather my data using baseline probes within training probes and maintenance probes, and how that fits into the single case design. So to give you a little bit of an idea on what that SCD for UVB training means, it's the how I'm using single case design for training speech language pathologists on the implementation of ultrasound visual biofeedback. The reason is because ultrasound imaging can be used for providing someone who has a speech sound disorder or is learning a novel sound because the person has it's bilingual, and let's say that it's learning English and French. With ultrasound visual biofeedback, if there is distortion on a sound, I can teach the person on where to put or how to shape the tongue in order to produce a novel sound, or in order to remediate a distortion that they have on a specific sound. And it was already written there. So, so in a nutshell, again, using ultrasound visual biofeedback for production of speech sound. You're good. And then what I did is that I provided training to the SLPs during 5 days. The reason why it was 5 days is because I wanted to, in order for you to implement ultrasound visual biofeedback for speech sound disorders, we have to think about the tongue as a muscle that is composed of different sections. So for each training day, I focus the training on a particular tongue section. In a nutshell, it'll be first day was just introduction, learning the ropes. So like teaching them how to interpret the ultrasound screen, how to interpret the what's the hyoid bone shadow, what are the parts of the tongue, that sort of thing. So it was on training day 2, 3, and 4, 2, 3, 4, and 5, the days where I targeted the specific tongue section. Specifically, first day I targeted tongue root, second day I targeted tongue dorsum, third day I targeted tongue blade, and then the last day we targeted tongue sides. Okay, so I did a multiple baseline design across participant and across behaviors, meaning because I had 4 dependent variables, that's why I did across behavior. So for each behavior or each independent variable, I'm looking at it in terms of each tongue section. So what I'm interested in seeing is, once I introduce the training, or once I introduce my independent variable, if I'm seeing changes in their knowledge or in their assessment of the configuration or positioning of each of those tongue sections. In other words, if they get better at making an assessment when it comes to tongue root, tongue dorsum, tongue blade, and tongue sides. On the right, you'll see my graphs. Maybe it's on the other slide that it will make a little bit more sense. Yes. So what I did is that you will notice that I have 2 participants. You will notice that each of the behaviors, I truncated the table, but each of those behaviors, because I staggered the introduction of my independent variable, you will see that each of them will have a different amount of baseline. So if you remember from Dr. Zhang's talk on single case design, baseline is another word, or it's another term for your control condition. So as I was, before I provided the first day of treatment, I gathered baseline data on all my 4 dependent variables, or all 4 tongue sections. Then as I introduced the training, I also measured changes in my dependent variable across those 4 tongue sections. And as I continue providing training to new tongue sections, I was measuring if what they learned for that tongue section that we learned about like the day before or 2 days before, if they maintain that what they learned. So for example, let's say that today you are gonna be my guinea pigs and I'm going to train you on ultrasound visual biofeedback. Today I'm gonna train you about tongue read. So before you came today, we did some baseline data and I noticed that none of you know anything about that, which is good. Today I give you training. At the end of this training, I'm going to do probes to see if you learned about tongue root. But at the same time, I'm going to also give you probes for tongue dorsum, tongue blade, and tongue side to see if you're still have no knowledge on those other tongue sections. Then tomorrow when you come, I'll give you the next tongue section. I'll give you other probes. And so because today we train tongue root, hopefully tomorrow you will have retained what you learned from today. So that's why I'm using baseline probes, within training probes, and maintenance probes. So when you're looking at the graph, every circle is the baseline probe. The square ones is for the within training probe or changes in the dependent variable immediately after training. And then the triangle ones are for maintenance to see if you're keeping what you learned. So how did I measure my dependent variable? I know this is a picture, but we didn't do it with just a still picture. I provided them with a video clip of a child producing a word. And then based on that, they needed to make a judgment on the positioning or configuration of the different tongue sections. We did it in a tongue section at a time. So it wasn't like with just looking at just one video clip, they needed to make an assessment for each tongue section. It was more like if today is tongue root, you make an assessment on the positioning of the tongue root, on the production of that word, and then that's it. Then we move on to the next clip. So that's how I measured changes. They needed to answer these two questions, and in order for them to have the full point, they needed to have both answers for both of the questions correctly. If they had both of them wrong or one wrong and one right, they still got a 0. I just made it harder for them, right? So lessons learned. Because I was measuring 4 dependent variables, I quickly found out that it can be a mess of a thing as you're monitoring baseline within training and also then the maintenance. So you would start looking like that bunny, how I looked the first time I was collecting data. I was like, what can go wrong? What can go wrong? Well, here's the thing. Here's the thing. My suggestion for you is that create a day-by-day master schedule of every single thing that you have to do for data collection. What I did is that I numbered all the probes, the maintenance and the within training on the baseline, and I created a master schedule for each participant each day. I created these small packets, so that way all I had to do is just take that participants folder, and I had in that folder every single probe that I needed to do for that day. And then next day, I took the other one and so on. So if the more dependent variables you have, my suggestion for you is create a master schedule, because otherwise you will be pulling your hair out because you forgot to do a baseline, or you might have forgotten to do a maintenance probe. And it's not the end of the world. It's not the end of the world if you lose that data, but since we want to see the functional relationship, the less data that you lose, then the better. And I think that's it.
Okay. So thank you to all of our presenters. Round of applause. Yay. Okay, I'm just gonna ask people to talk loud and turn that off, because hopefully what we're gonna do now is engage in some conversation. Let me pull this back. Okay. So, I'm sure you've noticed that this presentation was much quicker and we had our panelists go through their study in much less detail than they might have if they had an hour to talk about their study. But part of the point of this is for us to consider the design and have some time for discussion. So I want to open up the room to our panel so that anybody in the audience here or virtually, maybe via chat, can ask our panelists questions, and we can start to discuss, like, how does this design work across many different ideas and many different disciplines. So with that, does anybody have, oh, I think I see T. Tyler raised his hand. Would you like to unmute, or do you want to?
Oh yeah, sorry. Forgive me, I'm just eating lunch, so I'm doing everyone a favor by staying off camera. Okay. But my question is, and thank you all for the presentations, very meaningful. Thank you for sharing your work. My question that I think I was intrigued by, which I'm open to any of you answering, is as you were describing at the very beginning, I think Emily spoke about this a little bit. Sometimes you'll be working on something where, you know, you'll be doing a case analysis, but sometimes when you submit your work, if people don't understand exactly what you're doing, like reviewers, you might have to translate that to them, you know, to articulate that. And I'd be curious to, if anyone's ever had that, like as a qualitative researcher, there's plenty of times when I've submitted something and someone had questions and I had to really clarify exactly what I'm doing so they didn't confuse it with something else. And I wonder if anyone's faced that challenge and how you addressed it.
So when I submitted this particular paper, I consulted with Emily, and in the design, so I had a design section of the paper, and I went into pretty good detail because in our field in particular, single case is often, probably like 3 or 4 years ago, was like, oh, that's a case study, and they didn't really understand the rigor behind a single case design. And now I think people are looking for particular things, or they just don't quite understand, well, how can you say that it was an effect? So I went into a lot of detail in this paper, and then even went into a little bit more detail to describe the participant versus the behavior and that interaction between the two. So I made a point, and we'll see what, I haven't gotten reviewer feedback yet to see if they still don't understand or not, but I wanted to try my best not to get that kind of feedback. So I put it into the paper.
And even, because it has happened to me before, even if I give that kind of detail, there's always the chance that a particular reviewer, which it's not familiarized or familiar with this methodology, they might come back and say something like, hey, you need to, like, for example, hey, you have only 2 participants. So your, not the validity of the study, but that should be a limitation. That's something that should be addressed. And my go-to is that, my go-to response is that I tell them or remind them nicely, so that way it gets published, that unlike group design, which it does, where the how big your sample size is impacts negatively or in a positive way your results. In single case design, you want to see the functional relationship, you know, across your dependent and independent variables. So it doesn't matter if you have 100 participants or 3 participants or 2 participants, because what you want is a functional relationship. You can make it the argument of, you know, what you're saying that it's a limitation, it's not really, because this is the purpose of this methodology, not what you think it should be. I don't know if I just went off on a tangent, but yes, in our field, I've gotten pushback because of it is considered a lower level of evidence, And I wonder if it's the name of the methodology, single case design, and people think, oh, it's just one person, one participant. Then it's lower level of when you compare it to the other levels of evidence.
And to give anybody who maybe is less familiar with this design some orientation to its history, it's a design that was popularized by special education research. And so it was a design born of a lot of minds coming together to think about how you collect data on populations you just don't have that many of. I think even about Dr. King's population, like clearly we can't go give people concussions so that we have, you know, enough controlled data to work with. But if we have a design that does work for a fewer number of people, but still has really rigorous control, that potentially benefits everybody. And so now if you were to submit a single case design to, say, a special education journal, you would find reviewers who are familiar with it and very up to date on what rigorous methodology is. But as the design spreads to other fields, yeah, there's mixed reaction, I think.
Yeah, and that's just the lack of familiarity, right? Of, you know, it's not your traditional kind of approach that we all work in within our respective fields. And so something's coming from the fringe and always takes a fair bit of time to start to get recognized and accepted. You know, the other part of that too is like making sure that the rigor is held up, and that when people start to put it in other domains, maybe like I tried to do, right, that we didn't follow all the standards. And so then that can lose its credibility a little bit.
But I liked your innovative approach of having your control data from other participants or having norms. So to me, that seems like that one of the wonderful things about this design is that it's very adaptable. And so it's almost like a hybrid where you can take norms, but then also collect some single case style data and still take advantage of that design.
You know, I looked at that and I thought, like, you could have just taken that line as your baseline, and then what you're looking for is not data that goes up, but data that stays on that line, you know? So basically it is like a single case because you have a standard or a baseline.
Right. Yeah, and you know, if we were to shift it to how y'all's graphs are, like, you know, that's at the baseline, then do the concussion post test to see kind of the trajectory that happens from there, and it would look similar to what we had, what you all were showing on there. But yeah.
And helps you get around the fact that you couldn't collect baseline data and then give someone a concussion. See what happens. Other questions?
I might have one. So working with disease processes that are degenerative in nature, Dr. King, you mentioned about your Parkinson study. And so I'm learning this is new, this design is really new to me. My question would be, because I'm considering a possibility of using this method, when we do these multiple baselines, how does how does a disease process that is declining factor in when we have, you know, 1, 2, 3 cases that we're presenting? We have a baseline, but by the nature of their disease process, their functional change may be impacted by the disease itself. Is this still a design method that can be used and has been published in the past? And I guess I'd like a little bit of information on that if anybody has any.
Yeah, I guess for our project, some points in terms of the functional decline that comes along with that. We've set up that our baseline has to happen within 2 weeks. So we're really bringing them in repetitively over a short period of time, with the idea that hopefully there's not the changes that are going on in that time period. And then once we get that, the intervention happens, and then we're hoping to stave off that decline with the intervention, but trying to keep it within a short time frame, I think, helps address any of the natural decline that comes with that neurodegenerative disease. We're also doing more than just 1 or 2, so then we can start to look at the variations and the reliability of that as a way to start to get some control baseline data for that population. And that's one of our plans with the data that we're collecting right now, is having some sort of reliability, variability metric within those baseline conditions to really characterize what might be going on within that Parkinson's population. Follow that?
Yeah, I think so. Yeah, because I was just envisioning that this idea that I could run someone who was actually getting the treatment, but could I still have a control somehow that is not getting the treatment, but maybe further out into remains, I guess their start point would be farther out. I don't know, I'm thinking through how we can alter the design to accommodate for that disease progression.
So my opinion on this design would be that yes, you could do that, because there are a couple of different places that you can build in controls to threats to internal validity. So you could look across behaviors for an individual, depending on what you were looking at. And that way you might be able to tease out what's the effect of the disease if all behaviors decline equally versus what's the effect of the intervention. You might also, in going across participants and having those variable length baselines, be able to keep some folks in baseline while you've started the intervention for others. So you're controlling for that time effect. And then eventually you do enter everybody into intervention in a multiple baseline design, but you could extend longer. The other thing related to single case design, which I think is not as well known perhaps as it should be. Clearly, this is a pet topic of mine too, so I'll throw it back to the panel in a sec. There are different designs that address different kinds of behaviors. So you have learned behaviors that, like, more or less once changed, stay changed, like learning to read. And you have behaviors that are reversible. So like reversible versus irreversible behaviors. So I'm picturing that something like, I don't know how much you're paying attention in class on a given day for the students that we teach. That's potentially a reversible behavior. Like one day might be good, the other day might be bad. The days probably don't affect each other that much. So the designs that you see here are all related to irreversible behaviors. But there are other designs that capture reversible behaviors if you expect there to be day-to-day variability. And so depending on what you want to measure, there might be some other designs out there that would better capture what you're trying to capture.
Thank you.
Yeah, I've seen some designs like that where you have the baseline, your treatment, and then you withdraw the treatment and you continue measuring and see if it goes back toward baseline, and then bring in the measurement, the treatment again. And so you're then kind of identifying that effect a little bit more.
And that's exactly it. It's the withdrawal and the reversal designs that, yeah, are for those reversible behaviors.
Yeah, one of the early papers I was reading into this for our project had exactly that idea of one cohort that does a short amount of baseline, another cohort that's a little bit extended, a third that does that. So, and again, you're really looking at the change once the intervention happens within there. I can kind of track down that paper and share it with y'all if you want. It was helpful for me, and it's a good visual to kind of grasp what the design looks like. We weren't hoping to collect that many participants to have a couple different cohort groups, but that helped address the concern that you're raising.
My question to the panel, then, kind of in this vein, is do you have any resources that are like your go-to either in responding to reviewers or learning more about the design that you'd recommend?
So there is I think it's out of IES, the paper that I always use is like they put out a paper that's basically standards of single case design, and it reviews all the types of design, and then it tells you very specifically like how this many data points, and I can share that. I always think everybody knows about that paper, but And then in the early, I think it's early childhood, child experimental, some journal, they put out a series of studies. I think even ASHA now, they've published one. I don't know how good it is because I usually reference those other studies. And I'm not gonna try to
Oh,
Prado, that's his last name.
It's the first author. And I think Dr. Zhang had that linked to, if you go to any of the information from that Lunch and Learn, she had that paper referenced.
Other things that I'll plug that you would want to make sure that you do is definitely procedural fidelity of your intervention day to day. I didn't do it for this paper, but another thing that I learned is to have a blind reviewer of your data. So like somebody who doesn't know, like you don't put the lines or anything in it, and you ask them whether they think this is a stable baseline or not, or do they think there is an effect or not. So it's somebody not, because this is all based on visual analysis, some numbers that you can report, but it's good to have somebody who is unfamiliar with your study and you don't give them a lot of very specific information on the graphs and ask them to judge. And then definitely I would just caution, like try to recruit more than what the 3, which is like the necessary number. So I always strive to say like at least 5 participants so that if there isn't an effect of one participant, you still have 2 others to rely on because you need to show an effect across 3 to determine the functional. And then I don't know whether this is relevant. I was just thinking you could have a behavior, a control behavior. So a behavior that you don't think is going to change due to the intervention, and that could maybe see some decline or take an account for that decline. I don't know. I was just thinking about that. Did that answer your question?
Yes. Yeah, I think those are some great resources.
They're also, I don't know if they're offering it, but I applied to IES. They had a single case training and it was like 4 days long and it was really great. And it was intensive all day for 3 days, but I left knowing what single case design is, and that's why I have always 5 data points in my baseline.
As a side note on that training, that it's the Institute of Education Science that they offer yearly. They have a rotating schedule where they do randomized control trial training across the 3 days, single case design across the 3 days. And I want to say maybe like that regression discontinuity design is one of the ones that they do as well. That sounds fancy. So yeah, it's something to look out for. They all have some good training if you want to learn.
Some additional resources. I don't know if it's the same article. I know it's from the WCW,
That's
Blood
it.
Work Clearinghouse. Is it the same one?
Yeah.
Okay. I also have it's borrowed from Dr. Lund, but she lent me a book dedicated to single case design, which is wonderful. If you want, I can send that information to her, and then she could we'll like share it. And also there is, I don't remember the name, but there's a calculator, because in single case design you have to use different stats. You don't use the group design ones. There are a couple of calculators online that help you with the PND, which is an effect size, or the tau u, and it's very simple. You just plug in the numbers and it'll tell you the effect size. I'll look for those links and send them to Dr. Lund.
What was the one that we were talking about? Hedges G.
That's
Is
like
there
for
a
the
calculator? I didn't report Hedges G, and I'm waiting to see.
I had to learn how to
yours.
calculate for a study Dr. Rivera and I were doing. So I know how to calculate that now
Yeah.
after hours.
And I that's not part of the standard, which is that What Works Clearinghouse is reporting these numerical things. I think this is newer and how it's shifting across disciplines. So I didn't report those things, and I'm hoping the reviewers don't. I mean, but if they ask me to, I absolutely will, and we'll learn, and you'll get an email.
Yeah. That's why we did ours, because we got a review back that asked for that.
Asked
And
for
so
it,
then we had to
so
learn how to do it.
I was banking on maybe they wouldn't ask.
Do we have other questions? I think we've got time maybe for one more. Okay, I have one. What made you guys decide to jump into this design? Adam, you addressed it a little bit, but like what,
For me, it was having a breather from all of these sometimes overwhelming, typical group design statistical approaches. Sometimes I would open SPSS and I'm like, I don't remember where to plug in this or that. But because it's been a year since I used it, and I tend to forget things pretty quickly. Something that from single case design that drew me in was that I didn't have to sort of like go back to the books and sort of like review, because the effect size stuff, it's pretty easy. And also for some reason, I really enjoy the time I need to spend on planning where I'm giving baselines and how I'm like doing that day by day data stuff, like planning how I'm going to collect it. I really enjoy it. So that's one of the reasons why it might not work for you, but for me, it's one of the reasons why single case design drew me in.
Yeah, I think I'll stick with my opportunity in terms of moving to a population group that is of interest to me in understanding the neuromotor components that go along with that. We're focused on Parkinson's right now, but there are a couple other avenues that I might be able to explore at this point in my career, and this is a way to get into that versus, maybe the alternative is finding a collective collaborative that's doing research around there and trying to tackle bigger design things. This gives me a little more control, more freedom in terms of where I might be able to move my research questions around those groups.
To piggyback on that, the parent reading training was not something that we found in the literature. Yes, the parent model was available, but not looking at teaching parents these literacy skills. So in terms of it being new, spending a lot of resources on a group design, I would have had, it's more time intensive on a study that I didn't know would have the benefit. So I think one great thing about single case is that it's only a couple of participants. So it's time, resources, money, resources. You're not having to spend as much, especially if it's like a newer, like pilot study. So that was great. Recruitment is hard, especially, kids with learning disabilities, but then also having like an intervention study that is time intensive for parents, asking them to come twice a week for an hour. Even on Zoom sometimes was hard for, especially the first parent-child diet. So I think in terms of recruitment, it helps to, get at least a couple at 5 is a lot better than having 25. And yeah, those are the two big ones for me. The hard thing about single case design, particularly related to my research, is for this study, it was nice because I was looking at parent behavior. So we took videos, Miguel and his research assistants coded the videos. But if I was going to look at something like grammar, comprehension or something, it's the testing of every session you have to be careful of, because you're taking data during every session. So that would be probably one of the hardest things about single case is if you're looking beyond behavior, you have to really get creative about the number of items you have for your assessment. And
I will say too, just kind of to round us out, I've recently submitted single case design as pilot data as part of an R01 NIH study. Nobody came back to me and said it wasn't valid. They were completely fine with single case design as pilot data. They didn't like other things about the study, but it wasn't my preliminary data that they had a problem with. So, for what that's worth, I think this is a really unique design that can be helpful in moving us forward. So with that, we'll end our panel presentation today. Thank you to our presenters. And I do want to plug, before everybody leaves, let me get you all right here. Our next panel presentation will be November 12th, and we'll deal with the scholarship of teaching and learning. So how do you measure studies that maybe are based around things that we're doing every day? And one of the reasons we're addressing that as a topic is because the RCAF award now has a topic area for scholarship of teaching and learning, where you could apply for funds to measure teaching and learning outcomes. Last year, the first year it was open, nobody applied. So I'm hopeful that maybe this next year in Harris College will, forge that path and have some people who I know do scholarship of teaching and learning work, seeking out resources to kind of grow that area. So with that, thank you, everybody.
Thank you.
With me, I don't know how often the parent, I wish there was like a standard behavior that you can always reference, because the baseline is always the hardest. It's not like, oh, no, we're not gonna do anything. Yeah, I thought that was really cool about that.
Interested in-